Okay, welcome everyone. I'm glad you could join us tonight. So this is the last session of the 2021 VLAP workshop. We're gonna wait a few more seconds here to let some more people join, and then we will go ahead and get started. All right, so we'll just give it another few seconds. Almost everyone is here, about half of you are here so far. So again, welcome, welcome, welcome to the last session of the 2021 VLAP workshop. So tonight we're going to have a cyanobacteria refresher presented by Amanda McQuaid. She is our harmful algal and cyanobacteria bloom program coordinator. So while you're waiting and watching tonight, you can feel free to introduce yourself or share a favorite VLAP memory in the chat box. You can also share a favorite VLAP memory in the group card celebrating VLAP's 35th anniversary. So if you'd like to contribute to that, the link is on this slide, but you have until the end of July to do that. So next, I'll just share with you a little bit of housekeeping. If I can get to the next slide. There we go, maybe. There we go. All right, so you are all muted and your webcams are not shared. So you can just sit back and relax, do whatever you would like tonight because we can't see you. So this session is also being recorded. It will be available at a later date on the VLAP YouTube page. If you have any questions, feel free to throw those in the chat box. Let me see if I can find the chat box and show you. Or maybe not. Okay, maybe I'll stop trying to draw. Okay, so anyway, the chat box is on the right-hand side in your control panel. It's sort of at the bottom. There's a little drop-down menu, so feel free to throw any questions in the chat box. We'll answer those at the end of the presentation. If you have any audio issues, there is also on the right-hand side a little audio arrow. So that drop down, you can use your computer audio, but if you're having issues with that, you can go ahead and use your phone audio. That requires a 4-digit PIN number, which you should have received when you joined the webinar. As far as anything else, that should be about it to cover the control panel and what you should use. If you want to hide the control panel, there's a little arrow at the top. So if you click on that, it should go away. So, well, apparently I'm still in drawing mode. I think I'd figure this one out by now. All right, so without further ado, we are going to start. I'm going to hand things over to Amanda McQuaid, and just one second. All right, Amanda, take it away.
Okay, thank you, Sarah. Thank you all for attending this evening's seminar on harmful algal blooms. Again, my name is Amanda McQuaid, and this is my contact. That cell phone number is a direct number for the cyanobacteria hotline, so we encourage you to take photos and send your location to that number. And I am trying to advance my screen, but it's not doing that. Hold on one second. Sarah, is it? Oh, here we go. I got it. Sorry, everybody. I'm kind of new to this platform. Okay. Hopefully you can see my next slide here, which is something you're probably all very familiar with. Plankton, which are microscopic organisms and particles drifting or floating in the sea or fresh water, is a major part of the sample collection that VLAB participates in. What we can tell from plankton toes is that there's a lot of variety of both plants and animals and bacteria in the water. And this changes seasonally, changes by lake, but there's just a whole other world in the water. And so using a plankton net, we're able to really concentrate what's in the water and take a better look. And I think that for the record, VLAB has participated in this for several years. It's given us a great indication of what is dominant in each of these lakes. So phytoplankton are microscopic plants. Sometimes this group includes photosynthetic bacteria, such as the cyanobacteria, but there's a great diversity of different types of microscopic plants in the water. And sometimes we see diatoms dominant in the winter time. We might see golden browns that emerge in the spring. We see dinoflagellates, sometimes indicates water quality. Of course, a very large group is the green alga, the chlorophyta, which can also create blooms. And then, of course, there's cyanobacteria, which is what we'll focus on tonight. But there's just a whole list of different types of organisms that can make a water look less clear, look a little bit green, look a little bit brown. And this is all due to these microscopic organisms. So one of the parameters we use to classify a lake, and I know most of you are probably familiar with the eutrophication process, but we classify our lakes in this classification based on the amount of phytoplankton, the amount of chlorophyll, the water clarity, nutrients, all of these different parameters that you collect with VLAB contribute to the classification of these lakes. And typically, a lake that is going through eutrophication is becoming more productive, more organisms growing, more chlorophyll, and that is sometimes associated with blooms. So what researchers are declaring is that more nutrients such as phosphorus and warming temperatures are like the key recipe for cyanobacteria growth. But there's a lot of other factors involved that might be playing into increased abundance of these cyanobacteria blooms. Not only temperature, but lake stratification is becoming a very hot topic in terms of oxygen and what it does for the release of nutrients into the lake. There's also some questions about lakes and their pH and what would support the growth of blooms. We are seeing that rainfall can trigger blooms, extreme weather events like big storms, and of course nutrients, which are a bit more complicated than just more phosphorus. I know you guys have probably seen this with Sarah. She presented this earlier in the series, but what we know is that there is a climate change occurring. The United States is much warmer, the entire planet is getting warmer, and this will only support warmer lakes and more nutrients and, of course, more blooms. So what are we talking about here? This is cyanobacteria. Some people don't like to use the word cyanobacteria, and I understand. They're formally known as blue-green algae, and it's okay to call them blue-green algae. They're not truly algae, they're photosynthetic bacteria. They have been around for, as long as the Earth has been here, and there's thousands of species and hundreds of toxins that we're learning about more and more every day. they're not just found in freshwater environments, they're ubiquitous in the environment, meaning they could be found in the oceans, in the soil, in the air, growing on rocks. So they're not just like a freshwater problem, but they're also not just an issue for any location on the earth. They're a problem worldwide. So the reason why we care, and there's a lot of words here, I'm sorry, but cyanotoxins are complicated, and this is not even a complete list of the toxins that might exist. Cyanotoxins range from the microcystins, which are very common. They target the liver, the digestive system, acutely can cause upset stomach, gastroenteritis, and chronically can create tumors. Nodularins are very similar to microcystins, but they're more common in brackish to marine environments. We have anatoxins, varieties of anatoxins that are neurotoxic and can cause seizures and acute death, especially common for dogs who ingest toxic cyanobacteria. There's saxitoxins, which are very similar to the paralytic shellfish poisoning or red tide. They can also occur in fresh water. They're also neurotoxic. Cylindrospermopsins are toxic to multiple organs, neurotoxic, genotoxic, and affects neurons and genes overall. Lingvia toxins, they can cause rashes, but also are tumor promoters. And then BMAA or DAB, these are non-protein bound amino acids that are neurotoxic, and chronic exposure may be linked to neurodegenerative diseases. So it's a pretty scary list of symptoms. However, this isn't even all of them, and not all of the cyanobacteria produce all of these, and that's what also makes them a bit tricky in understanding. What we are very concerned about, I think more so than anything, are dogs and children and people who can't, really make these decisions by looking at the water. Canines especially are vulnerable to consuming a lot of lake water, getting bloom material on their fur, grooming themselves, and then unfortunately, in some cases, dying from the exposure. There was actually an article in the news recently I should have included in Florida, West Palm Beach, a dog down there consumed like a mat of algae that was stuck on a lily pad and had severe liver hemorrhaging and died. These things happen more than we are aware of. It can be really difficult to tie these events to the source. But in a study by the CDC led by Lorraine Backer, they reviewed some cases over a few decades to really follow the canine intoxication with both marine and freshwater exposures. And typically, it was followed by freshwater cyanobacteria exposures where dogs were confirmed to become ill from cyanobacteria. What is also a bit concerning about blooms is that they're aesthetically unpleasing. It's not ideal to show up at the lake or the beach and have it covered in blue-green material or brown material or scum or dead fish. In this case, when the blooms are decaying, they actually take up oxygen, which is ultimately what will kill certain fish. We also have other stressors that cause some spawning stress and deaths. But we also can see that after blooms are decaying and dying, their oxygen is actually what is really causing these deaths. It's concerning, and it's also unpleasant to show up at the beach like this. But it's a huge variety of different things that are creating these colors and these scums. And it's the prolific growth of these microscopic cells that rise to the surface, and then we can see them. So they're growing in the lake, and we know that they're there, but then at times they are given a lot of nutrients from perhaps runoff or something internally feeding this stuff. They can outcompete other algae very, very quickly. But microscopically, they're very different. They're as different as plants. They have different requirements and different growth ranges. So in this case, we are seeing a lot of variety of different things out there. Don't assume that it's just pollen. We are seeing a lot of pollen right now, but what we're also seeing is sort of a mix of cyanobacteria blooms that are at the same buoyancy as a lot of the pollen, and it's sort of masking it. So look for any of these signs when you're out in the water. You have your typical blue-green surface scum that creates these like streaks in the water. We're also seeing this very orange colored bloom at a few lakes. That's a mixture of a Warrenicinia cyanobacteria and this green alga that looks pretty orange but is called Botryococcus. It's sticking with the Warrenicinia in this upper right photo and creating like this amorphous blob. So there's like a lot of different varieties with the cyanobacteria. It's not always blue-green. We also see some lakes that just turn green altogether and don't have the surface scum. We've seen benthic mats that created a black looking sludge that came to the surface and ended up on a beach. There's also these like blue-green marbles that you can find in the water. There's blue-green flecks. There's white clouds indicating the cell death. There's just so many varieties. And when these things are mixed with other algae or pollen, they can look a little bit strange, even if you are used to seeing cyanobacteria. You might question it. The other photo here, the red dots, is a whole other category that we cannot see. These are called picocyanobacteria. And in this photo, this is epifluorescence light, allowing those cells to shine. And that is due to the pigments that they contain. So we've talked about cyanobacteria toxicity, but not all cyanobacteria are toxic. Toxins can be mild. They can just cause skin irritations, eye and ear irritations, which, you know, can be annoying, but sort of is like a cold. You might not realize that you were even exposed. It's like very familiar. There's also gastroenteritis. You could go to the doctor and they'd say, oh, it's something you ate or drank. So these are kind of elusive symptoms that could be applied to a lot of different things. But what we know is that the great variety that we see have the potential to produce a whole range of different toxins. And some are even able to turn those on and off at convenient times that we don't understand. And the photo here are just the 4 most common types of cyanobacteria that we have documented in New Hampshire. The upper left is called Dolichospermum. It used to be called Anabaena, and we still like to call it Anabaena. But that one is the most common, and that's what we have been seeing in New Hampshire this last few weeks is all of this Dolichospermum. They can produce anatoxins. So if you remember Anabaena, remember that they produce anatoxins, and those are those acute nervous system failure toxins. Have also been given a nickname as fast death factor for the toxins they produce. These are the ones that we're worried about dogs consuming too much of acutely. The upper right is called ephenoxaminin. That's been known to produce neurotoxins, saxitoxins, anatoxins, and microcystins. So a huge variety could be associated with any one taxa. We also have Oscillatoria or Planktothrix, the bottom left photo there, where this bloom could be down in the lake, deep in the water column, just kind of hovering in the thermocline for the entire summer for years, and you will never see it. But sometimes we do see this come to the surface, typically towards the end of the season, maybe August, where these different organisms are allowed to sort of change their buoyancy because of the stratification of the lake and several other factors. But the point being is that these could be blooming really deep in the lake without even noticing it. The bottom right photo is really hard to see. This is a sort of a crude view of Microcystis, and this is the one that I worry about. Microcystis is most often toxic. It produces microcystins, and this is the one that can get toxic very quickly. It doesn't take too much of this organism to create a lot of toxins that the EPA would recommend not drinking or recreating in. We also have some pretty unique cyanobacteria for cleaner lakes like Winnipesaukee and Sunapee. Gliotrichia is one of these organisms of cyanobacteria that we do tend to find in the big, deeper, cleaner lakes. It's a low toxin producer, so for its size, it's a very, very large size relative to other cyanobacteria, produces a low level of microcystin, but we don't really know about the range of toxins that this organism can produce. It also doesn't really look like a surface scum. When you see it, it looks like these tiny little dots in the water, and you become more aware of how different this organism can look as a bloom when you see it. We also have nostoc in a few of our lakes. These are just a few of the lakes we have documented. There are more, Pawtuckaway Lake and Millen Pond more recently. There are these nostoc balls. They're like little blue-green marbles that do not create surface scums. You'll just kind of look through the water and see them on the bottom in the sand or in the mud. Within each of these balls, though, are tiny kinked coils of the cells, and these can be neurotoxic. We also have this group of benthic cyanobacteria. There's a whole range of different types in these mats, but Stygonematales is one that we're seeing in both Winnipesaukee and Spofford Lake. We don't know much about its toxicity. Some preliminary results indicate that it can produce a variety of toxins, and the consumption of that would be based on how much of this black material was consumed. So it's not necessarily toxic in the water, but the mat itself, which dogs again can be attracted to. We also are seeing a lot of non-cyanobacterial like blooms. Green filamentous algae is one of the biggest calls we get all summer, and we are seeing more of it. The Spirogyra especially is common, but there's a whole range of different types of green filamentous algae, and they sort of create like this like tumbleweed in the lake where you can see this big pillowy like cotton candy like bright lime green mass, and people wonder if it's cyanobacteria. So we do want to keep documenting this, so please let us know if you do see this, because it's seemingly more common, and it might be due to warming waters. We don't know a lot about why, but what you can do just to feel better about the situation is to try the stick test. So grab a stick, try to lift the mass. If it all kind of sticks together like a hairy sort of slimy strand that kind of sticks together like this in the picture, it's most likely just green filamentous algae. If you were to poke it with a stick and try to lift it, and it was a darker color, like almost black or really dark green, and it sort of clouded the water and was hard to lift up, that could be cyanobacteria. But even with the stick test, we want you to take photos and just send them to us so we can help you identify it very quickly this way. Last summer, we actually had a lot of Wolffia, which is just a tiny aquatic plant. It's also known as water meal or duck meal in some cases. And it's not harmful. It's just you wonder, this was the Nashua River, and people were reporting this throughout towns along the Nashua River, and Wolffia just did very well last summer. We also sometimes see a Euglena bloom. This did also happen last summer in the town of Meredith. This was not cyanobacteria. It looked just like it. It might be hard to tell in this photo, but what happens is this Euglena organism is sort of its own kind. It's like half animal, half plant. I know this is a little more complicated than that actually, but this organism creates spores that can create this sort of surface scum slick. And Euglena can also consume cyanobacteria, so they might kind of look like a bloom, and it's really important to report these two. So far, we don't know about the toxicity in New Hampshire. There is a certain species that has been documented that does produce ichthyotoxins, which are harmful to fish. So with what we do here at New Hampshire DES is we are just trying to let people know when we know that there are cyanobacteria blooms or elevated levels. we're really willing to do it in any way possible to just increase the awareness and let people know to be on the lookout. A lot of the times these blooms are just quick, sporadic events, but someone might approach the shoreline at the wrong time and not know about it. So our advisories are really just an alert system, and we've sort of modified the approach. This last year, because we were short-staffed, we came up with a different plan for responding to blooms. We also wanted to keep this plan to stick because oftentimes, again, we will get a call about a bloom and we'll drive a few hours out to find the bloom and it's gone by the time we get there. And that's great news. But what we want to do is increase the time efficiency in getting the word out as it happens. So officially, cyanobacteria advisories are issued when samples are collected, identified, and those concentrations exceed 70,000 cells per milliliter. So that is the premise for which we will issue an advisory. But what we are also trying to do is create local alerts where if there is a photo that is shared, we can confirm that right away and let people within the association or the neighborhood start spreading the word by just showing the photos. Because really that, with all the variety of different things we're seeing, that is the best way we think to let people know what to look out for. This is what's on this lake right now. It might go away. It might be just down at your neighbor's house. And so having this sense of community and communication is really important when an advisory or a cyanobacteria bloom alert is going on. We want to get everybody on board just understanding what it is, and not necessarily close beaches or close lakes, but just let people know to be on the lookout and to avoid it when you can see it. Unfortunately, sometimes lakes, the entire thing is filled, and that can also pose a danger. And so even lakes that might just look green overall or have just a hue of color might be indicating the start of a lake-wide bloom and not an isolated patch found in different locations. So blooms have been happening for decades and decades, hundreds of years really, but since 2003, New Hampshire DES has been keeping some track with the advisory process. And there have been over 300 publicly issued cyanobacteria related advisories, warnings, alerts, and that just kind of encompasses all the different terms we've used over the years. But overall, it's just a lookout system. Hey, there's been this spotting. Please be on the lookout. That's what the message is supposed to be. So in recent years, we have had the highest number of advisories in reaching past like the 30 summer threshold. And then this last summer, we had 23 advisories, but we also had 35 alerts. So totaling, close to 60 events last summer. We think that this is a sort of a product of increased public awareness, but also when we've had some extreme weather like the drought we had, we're seeing certain lakes having blooms that really weren't blooming before, but they were pretty short-lived. When we look at all of the data from since 2003, again, we are finding some of the same organisms, and Anabaena or Dolichospermum has been the most dominant. But we are seeing a lot of different variety and sometimes even multiple blooms. This is just the report for the most dominant per advisory. And when you look at those more common ones that we see in New Hampshire, this was modified from UNH's website. There are, a good group of really common ones that we see in New Hampshire and other states, of course. But here, what are the associated or known toxins for this variety? There's a lot, and this doesn't even really cover everything. But what you can kind of look down the list and see is microcystins over and over and over again. That is because it's the most well studied, but it also tends to be the one that most of the cyanobacteria are at least capable of producing. It's also a toxin that is known to genetically regulate its expression, so it can turn it on or off at any point without prediction. So one thing that we are trying to do at New Hampshire DES is test for cyanotoxins. We've been very focused on microcystins. That's because there are some health advisory levels recommended by the EPA for drinking water and for recreational contact. Again, it's the most common one that we see. It's also a very stable toxin that can last in the water even after a bloom has decayed. So we are using enzyme-linked immunosorbent assay, or ELISA methods, which is just a kit that you can buy from a company, run the assay, and get an idea of how toxic by microcystin toxins a sample might be. We've tested this quite a bit. I have experience with this from my own research, but also just at DES, we've really focused on microcystins, but we're also trying to get into some other types of toxins as well. But from these results from last summer, we're not testing all of the samples that we get. We did collect over 500 samples related to cyanobacteria complaints last summer, and about half of those were tested for microcystins. Of that subsample, of those 50% or so, there was only roughly 20% that had positive results for microcystins, and only 3% were above the test limits, which also exceeded the EPA recreational guidelines of 8 parts per billion. So this is a very low incident rate for microcystin toxicity, but this is a very rough way of looking at this. This is promising in a lot of ways because a lot of the levels were below detectable levels, below drinking water levels, but this is occurring unpredictably. There's a lot of different contradicting, lots of different types of toxins that we're not even able to test. So I would never want to say, oh, look at this, it was not toxic this amount of time. This is just a snapshot in time, and this is just one toxin, and it really doesn't cover the whole range of toxicity that can occur in a bloom event. For example, your shoreline might look like it has a green ribbon, and then an hour later it looks like a whole green cloud is there. The concentration can change so rapidly. So any area that we are testing is, again, just sort of a snapshot in time to get a sense for the range of toxins that these blooms are creating, but it does not cover them all. So the ribbons of cyanobacteria really represent like a timeline for this accumulation. So again, like I just mentioned, like you could go out in the morning, there's just a green ribbon, and then you come back out and there's another ribbon. And this is sort of on a calm day can really show all the accumulation of stuff that's growing in the lake, rising to the surface, and then gently being blown to the shore where you can see it. And so throughout the day, this could increase or decrease depending on the wind, which is very unpredictable. This is constantly changing due to a lot of factors, and the toxicity may rapidly change as a result. So what should you do if you see a bloom? We still really want you to just take a photo, send your location, but first and foremost, please avoid contact, even if you think it's just pollen. Keep pets and children out of the water, especially who might be likely to consume this material. We, again, just want to document this because we will drive out and it will be gone, which is great. But again, we want to at least see this, the extent, the severity, and let you know right away what we think you're dealing with. And you can contact me directly on that phone number or by email. There's other things you can do, and VLAP has been involved in a lot of different parts in this cyanos.org, which is a EPA Region 1 monitoring program that DES has participated in, and VLAP especially has contributed a lot of samples over the years for this project. But basically there are 3 tiers to it, one being Bloom Watch. This is just like what we're all doing. We're all just sort of looking for blooms and looking to see if there's something to report and let people know about. There's also cyano monitoring, which is something that the VLAP program especially has helped with in previous years, using water sampling, integrated tube sampler to collect water routinely. So even if there's not a bloom, just collecting water to see the water over time, what's happening with cyanobacteria growth. This has been largely using fluorometers to determine concentration. So with the VLAP samples that we had run over the years, we were just finding that we were not finding anything. We were really we were finding that the blooms were sporadic and we were not catching them during routine monitoring. So the levels that we were finding were all just basically zeros. And so we didn't really have much to report. And you can look at that as a good thing as well. But these fluorometers they're not complicated, but they're really lake specific. In my opinion, you would want to use the same device in the same conditions to use this as a indicator for cyanobacteria growth. And when I've spoken with some people that have their own fluorometer and use it, they find it useful because they know what number to them means zero and what number to them means that there's some growth and activity. So if anyone is interested in sort of using that approach again, please contact me. And then finally, there's Cyanoscope, which is basically what I do. But if you were interested, you could also just, get your microscope and try to determine what you have from your sampling efforts. So the EPA cyanos.org is a really great website to check out. We really do promote Bloom Watch, but if there are any other tiers of this program that you're interested in, we would love to help you. So just shifting gears a bit about more like on what you might want to be involved in terms of research and cyanobacteria in New Hampshire. And I only have a little bit of detail here now, so if you're interested, we could talk another time. But I just wanted to present this. I call it the Haney Project. This is Jim Haney and his daughter Jessie Haney, and some others with UNH who are trying to do a pilot project using satellites and the fluorometry data from that EPA effort. So if you didn't know, there's satellites above us all the time, and we have different uses for satellites, of course, but the priority for this project was to sort of ground truth the satellites that are existing with data that we're collecting on the lake. The ultimate goal being, can we use satellites to predict or show us in real time the conditions of the water clarity and give us an idea if there is a growing cyanobacteria concern in water bodies using satellites. So for this, there's a lot of, good things about the satellite use, but there's also some setbacks. So for example, if the satellite goes over and it's a very cloudy day, you won't see anything on the ground or in the lake. But the project's premise is that with the combination of several satellites, there are dates in which they all fly over the lakes, and we could try to collect more water quality data at those times. And over, the summer, if we get enough data, we can use some of this fluorescent data to try to ground truth some of the satellites that are already in existence for New Hampshire. So the left picture here is just a very pixelated view of a lake where you can see sort of these pigments coming out in the satellite view. What we're trying to do is do a combination of the signals from the satellites and compare that with the phycocyanin fluorescent values that we have with our handheld fluorometer. Again, a component of the EPA cyanos.org approach. Preliminary results are a bit promising. There's, there's some different approaches here, which I'll try to walk through step by step. But basically, we're trying to use phycocyanin and different fractions of the water to predict what is already showing us on the satellite so that we can, again, sort of use this to say, yes, our fluorometric readings are useful for satellite uses. And so far, there's been some good correlations with the phycocyanin that we're collecting from the water and the wavelength that the satellites are operating at. And so there is a very quick protocol. Again, if anyone's interested in any volunteer efforts with monitoring, we can talk another time, but this pilot project for this summer is called the Satellite Integrated Cyanobacteria Monitoring Project by UNH or the Haney Group. And really, they're trying to make use of these different components of the EPA project. And it's really broken down into two things. It's collecting water and it's collecting plankton, the two things that you guys are probably very familiar with doing. The fractions that they are looking for are whole lake water, just a scoop of water from the lake with an integrated tube. Then there's also the less than 50 micron, so that's just water being passed through a 50 micron net. So you've split that water in half, basically, whole lake water with versus the less than 50 micron size. And then with the plankton net, you're really concentrating what's in the lake and looking at what they're calling the BFC. The BFC is the bloom forming cyanobacteria. So the approach we had before with the EPA was sort of a very dilute look at pigments because there wasn't that much in the water column. But this approach using a plankton net will now allow you to sort of create your own miniature bloom in a plankton net and be able to use that more often to predict the growth phase of these blooms. What this project will also contribute to, again, is the use of satellites for quick and real-time use of satellites for looking at cyanobacteria blooms, basically on a map. So that all of that might sound very overwhelming, but I just, again, wanted to introduce certain projects that are happening in New Hampshire that the EPA supports and that could be very useful for New Hampshire, but also revisiting some of the other approaches that the EPA has guided us on. And I think all you can really do is be on watch. You really just need to look before you leap. There's really no lake that doesn't have cyanobacteria at some point. Of course, there probably are a few, but it's a very common organism, and it's just we don't really see them that often. But when we do, we want to stay away, and we want to help spread the word by sharing photos. And so if you know that's the level that you're involved in, that's good enough. But there are these other opportunities, and I welcome you to contact me if these interest you. With that, I'll just thank you very much. Again, this is my contact information, and I will try to see any sort of questions you have in the chat box here. And I am not seeing any, but I might be in the wrong location.
I don't think so. I only saw one come through, I think.
Oh, say
Amanda.
when. Okay, I see something that says advice to swimmers and waders when blooms occur. Yes, my number one advice is to stay away. Make sure your dogs are put away during this time of uncertainty. If you think you were in it, just rinse off, wash off. Of course, try not to swallow the water, but if your pet went in, really make sure that their fur is washed off. Secondly, you could take a photo and text it to me so that we can just get the communication going so that we can say, okay, yeah, that's what it is, stay away. Let's get a sample and follow up that way. But really, we just want you to know what it is. We want you to understand what it looks like, just like you would want to know if there were venomous snakes in your area or, poison ivy even. It's just more of increased awareness. It doesn't mean that the lake is ruined or anything other than the fact that there was just a fast bloom growth, a fast occurrence of these particles that just did really well for some reason at that time. But of course, what it can also show is a trend towards eutrophication, and we are seeing more recurring issues for certain water bodies that have led us to investigate further and have really sparked interest locally for people to really pay attention to the watershed. So the blooms can also just sort of motivate a community to really look at your own shorelines and your own landscaping and any runoff that you can see, because really it's the people who live on the lake that know the lake the best. And if you're seeing anything that seems to be contributing excess nutrients or excess runoff, it's really great to identify those issues. Of course, you need a very strong local community involvement and support for these projects. But if you see a bloom, stay away, take a photo, record your location, and just let me know, and we can take it from there. You just really want to rinse off and avoid, you really just don't want to consume it. I mean, that's really what it comes down to. But unfortunately, you could be, affected in other ways: skin rashes, eye and ear irritations, some things that are more mild, but still, something you could avoid if you know what to look for. I think I see another question here. I don't know if you can see what I'm looking at right now, but I cannot really open the question all the way.
I can see it. Do you want me to read it?
Is there any correlation between water depth, multi-day warm temperatures above 85 degrees and alga and cyanobacteria blooms? Yes, there can be. This is really pretty complicated because there's cyanobacteria like all year round to some degree. But we are finding that warming water could really support the prolific growth, and that we're finding that some other factors play into this as well. Water flow and just, where does the bloom end up? Sometimes it just could present itself in one location of a lake because of the shape of the lake. But we are expecting more blooms with warming temperatures. I don't know if I covered that whole question. Let's see. Yeah, Sarah, can you see these better than I can? I don't know if you can see what I'm
I
looking at.
can.
You cannot
Yeah.
really like
I can,
open?
no, I can see them.
Okay.
So we have a question: how soon after a bloom is it safe to go back in the water?
Well, that's a good question. I mean, what we would do is if we had an advisory, we would follow up with resample to confirm that it's diminished, or at least no one's seeing it and we can't track it. So we would remove the advisory. And at that point, it's probably fine to go in the water. If you do see a bloom turn blue or white, like aqua and milky colored, you would really want to stand clear of that area for at least 24 hours, but there's really no state recommendations on that. We will remove an advisory when the cell count is lowered again, recognizing that it might very well just be at the end of the other end of the lake where we can't see it. But that's where we really rely on the people who live there to help us out to say, hey, are you really seeing it anymore? Are you not seeing it? But generally, it's fine to go in once the bloom subsides. There is this release of toxins into the water upon cell death. Some of the toxins will degrade very quickly. Some will last for days to weeks. And so I don't think that DES would ever recommend just not going in the water all summer because there could be microcystins still in the water. It's generally very low levels, but they can persist. So it's really important that you don't draw water from the lake for anything consumption at all, like any purposes of consumption. Even boiling is just not advised. But for recreational swimming, if it looks clear, it's likely okay. You would probably just be a little more careful to look where you're swimming, look to make sure it doesn't look turbid, meaning if there's any particles at all, you really want that water to look pretty clear when you know a bloom has just occurred.
Okay. So we have a question: are alerts listed on the DES website?
They are not listed on the DES website like our advisories are, so they're not something you would find on the map. We are sharing these in a weekly newsletter, and if you are part of the community, you would probably get an alert statement until we could collect samples and confirm that it is an advisory. Then it would go on the map. But what these alerts really are these blooms that seemingly just last a day. Everyone saw it. We tried to get a sample, we missed it, it's gone, no one's seeing it anymore. There's no real reason for us to issue an advisory, but we want people to still be on the lookout because a lot of these lakes are large. It doesn't really just affect one side of the lake. It's really by chance that you see them. Again, they're growing in the lake, they're coming up to the surface. they're caught in the surface tension, and then it's just wherever the wind may take them that you will see them. So again, our alerts are sort of just for the local community until we can confirm that it does need to be an advisory. It's really just based on the rule that we really need to collect a sample and confirm and count it first before we issue those advisories. They're really the same thing in a way. The alerts, the advisories, we're just trying to get people to be on the lookout for it. But when it's an advisory, it's lasting longer than, say, 24 hours. It's more persistent, and we want to make sure people know about it before they, go to the beach that weekend.
Okay, they're coming in now. So here we go. What's the next one? If there's a bloom on one side of the lake, will people or dogs be affected by swimming on the opposite side of the lake? So basically, do you need to be standing in the bloom and drinking it in order for it to be dangerous?
No, I would say like if you drive your boat out to the middle and it looks clear, it's probably fine. Again, if your dog is swimming in it and you really don't see much, it can accumulate in the fur. It's kind of like its own plankton net, if you will, where it's getting trapped in the fur. So rinse your dogs off really well. But no, it should be fine. You really are just looking for this accumulation. If you can see it, stay away from it. It's highly concentrated if you can see it.
Okay. We have a couple that are sort of related to you mentioning not using lake water for any sort of consumption purposes. So the first is, does reverse osmosis or ultraviolet light water filter systems, do they filter the cyanobacteria?
Yeah, the filtering process, like, and like a triple or 5-point system for reverse osmosis should do it. It should remove all of the particles. What is sometimes concerning directly, if you're sucking bloom water in, it will go through the filter, the cells will get trapped, but the toxin will not. The toxin will get through most filtration systems. And it's usually the microcystin, which is more stable, that would likely survive that process. More acutely though, if you were to suck in like a benthic mat and have a lot of that material just stuck on the filter and then that water is trying to pass through, you might acutely be introducing more toxins into the water. That has happened in other parts of the country in their finished drinking water to see high levels of antitoxin and microcystin especially. There's usually not just a simple UV treatment that will kill the toxins, although it just really takes time for them to degrade. There are uses of certain, basic water treatment processes, using potassium and alum and different copper sulfates and whatnot. And so it gets a little more complicated for a public water system. But for you to draw the water from the lake, it is risky for a lot of reasons. It does collect a lot of material, but there are little microscopic things that get through, including toxins. So during an active bloom, it'd probably be best not to use the water for consumption. I mean, we recommend at DES that you don't use the water for consumption at all. Again, don't boil it, but even still, you wouldn't really want to use bloom water even on your vegetable garden. You want to be really cautious during a bloom because there's a lot we don't know about how toxic it is in real time.
Well, that's a great transition to the next question, which is it safe to use lake water to water vegetable gardens? So,
Well,
yeah.
this is the thing. Yeah, we've been talking about like how we can advise people on like composting bloom material that's on your beach, for example. Like, can you just scoop it out and, compost it? It's probably going to be fine if it truly goes through composting, where you have a compost heap, you let it degrade. But there's also a lot of evidence that shows that if you were to directly irrigate your crops or your vegetables or your garden with the cyanobacteria bloom material, some of the toxins are able to accumulate in the fruits of these plants, fruits and vegetables mainly. but it can also be a very sticky substance on leafy vegetables. So like, you know, maybe acutely it'd be fine. Like you might not get a big dose by, you know, like doing, you know, watering your garden. But there are a lot of chronic effects with this stuff. So there's been just advice from various researchers who look at food web dynamics that suggests that some of the toxins might biomagnify, and if you were to practice that long term, you might, you know, suffer from some of the side effects of cyanotoxins, which again could be just like upset stomach, but could be as serious as neurological disorders. It is not recommended to irrigate with the cyanobacteria water, but there's no rules on that. So it's not like you absolutely can't do it, and probably will even help like some of your garden grow, but we would say it's just probably safer to avoid using it on anything you plan on consuming.
Okay, thank you. We have one last question I see here. And it's, is there a key temperature range or a tipping point in temperature in the meadowlimnion that could promote a rapid bloom? So this is for smaller ponds. He's asking about 15 to 50 acres and 5 feet to 50 feet in depth.
That's a very good question. I don't know if there is a specific tipping point identified in New Hampshire lakes. I know that there's some really good papers by Wayne Carmichael, who has a famous paper called Blooms Like It Hot. And he's really demonstrating that there is an ideal temperature for bloom growth. But it's not necessarily matching like the patterns we see with blooms here in New Hampshire waters, like in June. But I think approaching the 30 Celsius range is sort of like that ideal, like hot weather, warm water temperature that people have quoted. But again, you know, some of these will grow even in Arctic lakes. So they do just fine. It's just they do better when things are warm or warmer. And small lakes, I'll just add, are more vulnerable to this because they'll, you know, the growth will happen and fill up the lake faster, of course. There's not like the dilution factor here. It also has a lot to do with the water flow, the depth, of course, as you mentioned. All of these can place a lake in a more vulnerable position than another lake. And that can also depend on what's in the watershed. And not only for today, but decades ago, what happened in the watershed is also important. So
Okay.
the last one,
I think that's it. I think that's it.
I think I
So
finally.
thank you so much.
Well, thank you. Yeah, it's weird not seeing you all, but thank you so much for being here. And of course, again, just contact me if you have any more questions or are interested in any other monitoring approaches. This is tricky stuff. It can be fun, but it's also a little scary, but we're trying to kind of meet you in the middle on like what this stuff is and just let people know what to look out for. All right.
All right. Well, good night, everyone. Oh, wait, hold on. There's a one last. Oh, okay. A lot of thank yous are coming in. So thank you, Amanda. Have a good night, everyone. We'll see you soon this summer, hopefully.
Bye. Thank you.
Yes, PowerPoint will be available. It's being recorded.